All attendees are in listen-only mode.
Good morning or good afternoon, depending on where you're located, and thank you for joining us for what should be a very interesting webinar. My name is Lindsay East. I'm the Marketing Manager for Wyatt Technology, and I will host this event. Today you will hear two presentations discussing the automation of biologics formulation preparation and stability studies. Our speakers are Dr. Daniel Sohm, Principal Scientist and Director of Marketing for Wyatt Technology, and Dr. Russell Burge, Application Scientist for Freeslate. Time permitting, we will conclude today's webinar with a brief question and answer session. If you have a question for either of our presenters during the webinar, please use the questions feature located on the GoToWebinar control panel on the right side of your screen. The control panel is automatically collapsed upon opening the program, so if you don't see it, please look in the upper right corner of your screen for an orange arrow button and select it. This will open your control panel where you will be able to view the questions feature. Please type your message or question in the box and be sure to indicate which presenter you are addressing the question to. First, we will hear from Dr. Daniel Sohm, who's been with Wyatt for over 10 years, first in R&D and then in the marketing department. Prior to joining the scientific instrumentation world, his professional endeavors included the semiconductor and defense industries. Dan completed his undergraduate degree in physics at the Technion Israel Institute of Technology, his doctoral research in the Brown University Physics Department, and postdoctoral research at the Los Alamos National Lab and the Weizmann Institute of Science. So without further ado, please allow me to welcome Dr. Daniel Sohm.
Thank you, Lindsay, and good morning to everyone. I think we're going to have a pretty interesting webinar today, and for those of you familiar with Wyatt's dynamic light scattering instruments and in the formulation space, you'll hear later on from Russ about some advanced applications integrating our instruments. But for those of you who are perhaps not so familiar with dynamic light scattering, would like to understand how it can help you in formulation, that's my role here. And so I'll jump right into the presentation, where I'll discuss Wyatt's instrumentation for high throughput dynamic light scattering and how it's used in screening biotherapeutic formulations. So there are a few areas where dynamic light scattering really shines in drug product development, and those are the phases looking at developability of candidates after identifying several candidates for continued development, pre-formulation to identify with minimum sample volume and maximum speed, essentially, ways to go forward in your studies with formulations that work well, and then in final formulation. And the ways in dynamic light scattering is used include assessing gross aggregation, so what are the aggregates present, and as you'll see in dynamic light scattering, we're focusing on submicron aggregates. Looking at the propensity for aggregation through the colloidal stability and the conformational stability, usually in terms of thermal behavior. And finally, if you're working on high concentration proteins, dynamic light scattering is really a great way for screening viscosity of highly concentrated protein formulations rapidly and with minimal sample volume. So how does dynamic light scattering work? Normally, when you think of light scattering, you're looking for a really robust, steady signal, and our standard static light scattering, or MALS instrumentation, uses the average scattering intensity to measure molar mass directly from first principles. Dynamic light scattering actually looks at the noise rather than looking at the average signal. And the noise, or the fluctuations in the intensity of the scattered light, arise from Brownian motion. Now what happens is, as you can see in the cartoon on the right side here, the optical wave will impinge on scatterers in solution, and they will scatter light, and some of that light is scattered in the same direction from the two scatterers towards the detector. That light is arising from a coherent source, and therefore there's a definite phase relation between the light scattered from the two particles here. And depending on the distance between those particles and also on the angle at which the detector is placed, those two waves might arrive at the detector in phase, in which case they interfere constructively and you'll have a higher light scattering signal. Or if the particles shift a little bit in position, they might arrive out of phase at the detector, in which case you have destructive interference, and that will lead to a lower light scattering signal on the detector. So as these particles are jittering around in solution due to Brownian motion, the phases are jittering around too, and you'll see these rapid intensity fluctuations in the light scattering signal that look kind of like this. So the time scale here for these relatively large particles, which we could actually take videos of, is milliseconds, but for smaller particles, for instance, monomeric proteins with their aggregates, you're actually looking at fluctuations on the time scale of microseconds. So we have fast detectors that pick up these fluctuations, and then we apply a mathematical algorithm called autocorrelation analysis, and what that does essentially is determine the spectrum of rates of diffusion. So dynamic light scattering is measuring from first principles the rates of diffusion. Finally, we apply the Stokes-Einstein relation, shown here, to determine from the diffusion coefficient and the temperature and viscosity an effective measure of size known as the hydrodynamic radius. The hydrodynamic radius represents the size of a sphere that diffuses in solution at the same rate as the molecules or particles that you're looking at. And that results in the graph that we see here on the left side, which gives us percent intensity of the scattered light as a function of size. One of the beautiful things about dynamic light scattering is that even without a preliminary separation technique, you can get a rough idea of the distributions of sizes in solution. So you can have a single sample, shine the laser into it, and if that sample has multiple size particles present, you'll get a distribution of sizes. The resolution on size is not great. We'll discuss that a little bit later on, but as you can see in the graph here, we can apply analysis that gives us a distribution of sizes. And so then this, as applied in the DynaPro plate reader, which I'll bring up shortly, you can apply this analysis to all the different wells in a microwell plate and rapidly assess the presence of aggregation in those different samples rapidly. Further looking at the analysis, so you might have in your solution a monomodal and monodisperse set of particles. Monomodal means that they are within a size range which is not resolved by dynamic light scattering, so they appear as a single peak. And monodisperse, of course, means that they're all the same size, and so that peak appears narrow. If the particles are polydispersed, for instance, due to aggregation, you might have monomers, dimers, and trimers. That leads to a broadening of the peak, and we call that broadening polydispersity. So the output of this analysis is actually the average size, the RH, and the percent polydispersity. If you have a multimodal size distribution, that means that you have in solution two size ranges, a smaller size and a larger size range, for instance, shown here in the cartoon, and those are actually resolved in the analysis. And if those modes are amongst themselves also polydisperse, so some aggregation perhaps on the smaller ones and the larger particles might have a distribution of sizes, again that leads to broadening, and so we can determine for both sets of particle sizes the average radius as well as the polydispersity. So you can get a lot of information with basically a very brief and non-invasive measurement. All we do is shine a laser into the microwell plate and pick up the scattered signal. So it's non-invasive. There's no fluid handling involved. The DynaPro plate reader utilizes industry standard microwell plates, the same kind that are used in spectroscopic plate readers essentially, and moves the laser around between the wells, picks up the scattered light, and will give you a range of the size ranges between 0.5 and 1,000 nanometer in radius. In terms of sensitivity, it can go down to about 0.1 mg per mil of lysozyme. Lysozyme is a protein with a molecular weight of 14.4 kilodaltons, and the sensitivity scales with molecular weight. So for instance, an antibody with about 10 times the molecular weight will have about 1/10 the minimum concentration for sensitivity, so it would be 0.01 mg per mil of an antibody. Minimum sample volumes, of course, depend on the particular plate that you're using. So if you're using 1536 well plates, you can go down to as little as 4 microliters of sample. More typical would be 384, where you're using maybe 20 on average, or 10 if you want to push it. You can ramp the temperature of this plate in order to assess thermal behavior. For instance, the temperature of unfolding or the onset of aggregation. And regardless of having a wide range of samples in your plate, some of which may be very pure and some of which may be heavily aggregated or full of particulates, you don't have to worry about setting the laser intensity. It automatically attenuates by up to a factor of 1,000,000. The DynaPro plate reader comes with the Dynamics software, which includes an API to interface to robotic liquid handling systems, and this was utilized by Freeslate, and Russ will talk a little bit later on about how the DynaPro integrates into their platform. This is one of the really cool options if you're looking at formulation or other types of measurements where you really need to work on high throughput and run through many, many plates and take that plate, make a measurement of dynamic light scattering, perhaps move it on to a spectroscopic plate reader or some other plate-based system, perhaps a plate sampler connected to HPLC. They're all available. Another really neat application besides formulations for the DynaPro plate reader is simply screening solutions for any other plate-based technique. For instance, if you're doing high-throughput screening on surface plasmon resonance to assess binding, just to make sure that your solutions are clear and clean and pure, you can load that plate into the plate reader, have it run. It'll probably take an hour and a half, and then you'll have confidence in knowing which solutions are sufficiently pure to get good results in your binding screen. So if you wanted to run a high-throughput screen just to assess gross aggregation, we could load up our workhorse well plate to 3 to 4 wells with something like 5 different candidates, 5 different pH settings, 5 values of ionic strength, and do that in replicate as well as loading on some controls to make. It's always good to have controls for solid scientific results. And you can readily run through those with as little as 10 seconds per well, still get quite good data. And so that takes you about an hour and a half to run through the entire well. And for each of those wells, you will determine the polydispersity, which is essentially the amount of small aggregates, as well as the presence of the large aggregates, which we see over here at about 80 nanometer radius. So here's an example of characterizing aggregation for a formulation before and after lyophilization. And there are 2 different formulations, one with and one without sucrose. And you can see here before lyophilization, so the solution with sucrose has a little bit larger polydispersity, and that may be due to, for instance, small sucrose particles in the solution. It's always nice to have controls, so if you actually had the solution without proteins, you could get an estimate of that as well if those particulates are present. And then go through the lyophilization process and reconstitute what happened in terms of aggregation. So after reconstitution, we find the formation of these large aggregates at around 100 nanometers in radius. And in this case, the sample with the sucrose had larger aggregates, but you can see some really large aggregates in the sample without the sucrose. That's right up here. Even though these are actually very tiny quantities of aggregates. So light scattering intensity is proportional to molecular weight, and that means that if you go to something with a very large molecular weight, you're sensitive to the presence of a very small amount of it. When you get up to something on the order of 100 nanometers, you're looking at molecular weights which may be hundreds or thousands of monomers, and when you're up in the hundreds of nanometers, you're talking about many, many millions, if not more. So these are very large aggregates, and we're sensitive here to the presence of a very small amount of them. And that's what I show over here. This tiny amount that you see here is probably on the order of just a few particles in that well. The data can be converted to percent mass. Now, I'm not a fan of converting the data to percent mass because there are some assumptions involved regarding the density of the particles and assuming that they're all the same material. But if they are in fact aggregates of the protein, then they pretty much have the same density, and the conversion to percent mass is fairly reasonable. And this is determined in dynamic software. We can output for each of these conditions how much of the percent mass was present in each peak. And so, for instance, in the lyophilized PBS post-reconstitution, we can see that the peak intensity was only 82% of the total, whereas the percent mass was 99.6. So only 0.4% of the total mass is actually present in that tiny peak. Dynamic light scattering is also useful for measuring unfolding, even if there's no aggregation. This is an example of lysozyme. So lysozyme, under many conditions, is pretty nice, and actually you can unfold it and refold it multiple times without any aggregation going on. And so what you see here is a temperature scan, and here's the radius of the molecule. And as we go through the temperature over here, we can see it increase a little bit, so increases from 1.9 to about 2.3 nanometers. And then the temperature is held here, 90 degrees. Now these data were not taken in the plate reader because it only goes up to 85. It was taken in our other dynamic light scattering instrument, the Nanostar. And when you hold it at 90 degrees, the size is not changing, and that means that it's basically not aggregating. So you could do a scan and hold. You could go up to a particular temperature, and you can hold at that temperature and assess the rate of aggregation. That's one of the possibilities with dynamic light scattering. And lysozyme here does not aggregate, we can see clearly, and we can actually ramp the temperature back down, and it would nicely refold. And this shows the data that we get at all these intermediate points. Some nice sharp peaks down here at the lower temperature. There's a little bit of broadening, and that's because at any given temperature around unfolding, you'll have in equilibrium some folded and some unfolded material, and that will increase the width of the peak. All this shows up very nicely in the data. And these data agree well with calorimetry and CD, which also look at unfolding. This is perfectly reversible. You can cool it back down, goes back to 1.9. Here's another example of high-throughput screening where we're looking at similar information for some different protein formulations. And here you can see that the temperature was held at 70 degrees, and depending on the formulation, you got different rates of aggregation. So essentially over time, you get up to 70, and then you just hold, and the size increases, which is an indication of the increased aggregation. So at the early stages here, there's a small change in radius, and that corresponds to unfolding. Later on, we get to aggregation. I don't actually show in the graph here some additional data that you get from dynamic light scattering. That is the count rate. The count rate is static light scattering, essentially. And because static light scattering is proportional to molar mass, what we would see here is that where there's unfolding, the average molar mass does not change. And so there's a change in size, but no change in average molar mass. That indicates unfolding. And then later on, when you get to aggregation, the molar mass does increase. Therefore, the count rate will increase. And that will give us a nice indication of when we transition from unfolding to aggregation. Again, these data correlate very nicely with other techniques, such as differential scanning calorimetry. It's useful sometimes when you're looking at different formulations to understand the difference in the aggregation behavior and not just whether or not it aggregates. So this particular monoclonal antibody we looked at 2 different pHs and ramped the temperature up to 80 C. Under 1 pH, 9.5, actually the aggregation was not too bad. It formed a kind of a nice stable oligomer with an average radius of 8.5 nanometer. Monoclonal antibodies have a monomeric radius of about 5 or 5.5, and so this approximately corresponds to a duodecamer. Again, by looking at the count rate, we can assess the average increase in count rate increases several-fold here. If you were to look at the aggregation of this under this condition in a technique such as differential scanning calorimetry, perhaps even fluorimetry, you might not detect the aggregation because it doesn't crash out of solution, and so you really have no positive indication of what's happening. On the other hand, at a different pH, 8.5, the aggregation behavior is quite different, and here you see a population with an average RH of 73 nanometers and some really large particulates, about a micron in radius, and so this would actually be crashing out of solution, and this you would pick up with some other techniques. But the dynamic light scattering gives you all this information. It tells you about aggregation even without crashing out of solution, and the aggregation behavior, the rates of aggregation, and the different size distributions. So that's gross aggregation. If you're at an earlier stage and want to look at the propensity for aggregation, there are a couple of indicators of propensity for aggregation. One is called colloidal stability and the other is conformational stability. Colloidal stability is the attraction of molecules to like molecules due to charges, hydrophobic patches, other things that might be on the surface of the molecule. And the way that we assess it is by looking at the change in diffusion coefficient as a function of concentration. So if the diffusion coefficient decreases at increasing concentration, that tells us that larger particulates are forming, and therefore there's a self-association. If the diffusion coefficient decreases with concentration, which is shown in the graph here, that indicates that the molecules tend to repel each other and that they're nicely stable in solution. Mathematically, the translational diffusion coefficient can be represented as a simple series expansion. So we have d sub 0, that's the diffusion coefficient of an individual particle, and and then the first coefficient of concentration, d1, which is known in the literature also as k sub d, is the first order behavior representing the self-association or self-repulsion. It is related to the second viral coefficient, A2, otherwise known as B22, including some other terms that are due to hydrodynamic behavior. The nice thing about D1 versus measuring A2 is that it can be measured in micro-well plates very rapidly and very efficiently. Here's a relationship between D1 and A2 and the different nomenclatures. So you might see in the literature as lowercase sub d. I prefer to use D1 just so we don't confuse it with some other terms that look the same, some other parameters that look the same. And if D1 is positive, generally it's repulsive behavior. If it's negative, it's generally attractive behavior. So interesting study that we did here was to look at how D1 changes with temperature around the unfolding transition. And so here's that antibody that I showed you earlier. Here's D1 as a function of temperature. And you can see that until we get to the range where it begins unfolding and aggregating, it's fairly constant with temperature. The black trace here is the average RH. So here again, RH is stable until we get to some transition where it increases from about 5 nanometers to about 15 nanometers. And this region over here would be the onset of aggregation. Interestingly enough, D1 actually begins decreasing several degrees before the onset of aggregation. That means that as the molecules begin unfolding and exposing their hydrophobic cores, they become self-attractive, but not yet to the degree to which they aggregate. That self-attractive behavior is reflected in the decrease in D1. And finally, where the aggregation starts occurring, we have a negative maximum D1. That means there's the most self-association. As soon as that aggregation occurs, those hydrophobic cores will now be hidden from the solution and hidden from the other molecules in solution, and therefore those molecules become less self-attractive, and D1 increases further. So that's an interesting way to utilize dynamic light scattering to get some more in-depth information about the behavior of the molecules around their unfolding and aggregation transitions. Finally, dynamic light scattering can be used to assess viscosity. And in order to do that, what we're going to do is we're going to take, for instance, our highly concentrated protein solutions, put them into the wells, and spike into those wells some polystyrene beads of a known diameter or known radius. And we're going to invert the Stokes-Einstein equation. So normally, we measure the diffusion coefficient, we know the temperature, we know the viscosity, and we extract the radius or the size of the particulates. In this case, we know the size of the particulates, and we utilize this to extract the viscosity. Here's a study which was done to assess the correspondence of the viscosities measured by dynamic light scattering and by more standard rheological techniques. And you can see that there's a very nice correspondence. The circles and the squares are two different techniques. The square is the viscometer, and the circles are dynamic light scattering. And these are two different protein solutions, highly concentrated, going up to 140 mg per mil, and still we have some very nice correspondence. So, I would not put this forth as the absolute best way to measure viscosity, but if you need to use very little sample, just 10 or 20 microliters of sample per measurement, and do it rapidly, and perhaps at the same time assess some other properties of the solution, they can just be loaded into the plate loaded into the plate reader, and the measurement is done very quickly. So finally, if you are working on screening biologic formulations, the DynaPro Plate Reader 2 is a really important tool. It assesses aggregation as well as propensity for aggregation through the colloidal and thermal stability, working on highly concentrated proteins, so you can also use it for viscosity. If you're interested in learning more, I invite you to visit our website. We have some mnemonic URLs that you can use. If you ask for the slides, we can send them to you, and you can just click on them. They'll bring you to our websites to learn more about dynamic light scattering, about the plate reader, and there are additional on-demand webinars about dynamic light scattering and other technologies that Wyatt offers, which you're welcome to view, as well as to read the application notes. And with that, I'd like to thank you for attention and turn it back to Lindsay in order to continue with the rest of our webinar.
Great. Thank you so much, Dan. Our next presenter is Dr. Russell Burge from Freeslate. Russell earned a PhD in molecular biology and biochemistry from the Scripps Research Institute in La Jolla, California. Russell received postdoctoral training at the University of Colorado in Boulder, where he worked on the biophysical characterization of RNA and DNA aptamers relevant to the biopharmaceutical industry. Russell's industry experience includes working as a scientist at KBI Biopharma Incorporated, where he contributed to over a dozen biopharmaceutical development projects. Today, he is an application scientist at Freeslate, where he designs and performs demonstrations of automation systems used in the research and development of pharmaceuticals. Russell's additional contributions to Freeslate automation range from market research to new product development. It's my pleasure now to welcome Dr. Russell Burge.
Thank you for that introduction. So today I'll be talking about application of automation for the preparation and stability studies of biologics formulations. This is sort of a logical continuation of what Dr. Dan Sum was talking about earlier. In this case, we'll actually be using the Wyatt DynaPro in addition to some other third-party analytics as part of larger workflows. So I wanted to give an introduction about Freeslate generally. We are a company based in the United States that we make integrated automation solutions specifically for drug development, both large molecule and small molecule. Today I'll be talking about biologics, and where we start from is using proprietary automation platforms. These are configurable automation systems. These are hardware systems with liquid handling, solid dispensing, and our own instrumentation for drug development. We also have our own high-throughput software that manages these systems and third-party software as well, and it's called the Lab Execution Analysis software, and this is a comprehensive data integration, data design, analysis, and reporting suite, which I'll talk a little bit more about later. And we also work together with our customers to understand the different challenges that drug developers face and the different benefits that automation can provide. So we know that drug formulation, especially for biologics, is challenging. We know that we have to create ideal biologics formulations that are safe and effective. They are robust, meaning that they maintain long-term stability at 2 to 4 degrees refrigerated temperatures for about 2 years. They need to have well-designed modes of delivery to the patient, and the formulations have to be compatible with those. Obviously, we have to meet regulatory requirements, and it is also advantageous for the formulations to enhance drug manufacturability. So here's an overview of how drug formulation development activities are working as part of these development programs. You can see at the top there's discovery research, which then transitions to preclinical and clinical research. And as you can see in the middle there, There are initial formulations and commercial formulations, and on the far left side of that we have the preliminary formulation studies. Nowadays there are companies that are even doing preliminary formulation studies as part of developability, which is nestled somewhere between the discovery research and the preclinical research. So we start to see formulation studies very early on, and they continue throughout the initial formulation. Once the dosage information starts to become clear from the phase 2 trials and phase 1 trials, we start to transition to the commercial formulations. And then there are oftentimes cases where those materials can be reformulated later for second generation or other indications. So as part of that, we have made automation that addresses these challenges. One of the areas where automation can provide benefits is improving study results. Some of these studies are complicated and require many different people to work on them and can be highly manual. So in this particular case with the automation, we're able to produce highly reproducible results because of how we can calibrate our systems in the beginning of a test, in the middle of the test if you need, and even at the end to verify the system is working as stipulated. We also, I'll talk a little bit today about how the automation can be used to alleviate subjective interpretations of certain results such as color, visible particles, and turbidity. And manual handling and labeling errors are virtually eliminated because the systems can be loaded up by the drug development scientists and the research associates, and the system actually takes into account all the places on the deck, everything that is done to the different vials and microtiter plates, and we also have barcode scanning and labeling capabilities as well. One thing we see is significant productivity improvement over manual performance. This enables a broad range of conditions to be characterized. This enables the drug development scientists to implement true quality by design strategies, because now you can take your design of experiments, lists of different samples and experiments you need to run, and the system will be able to perform those, and it will allow the scientists to spend their time on more valuable aspects of the study. And part of the way we do this is we obviously use the automation to do things that would normally be manual, but we also have novel automated methods such as visual inspection, pH, and viscometry. In today's talk, I won't talk too much about those, but they will be highlighted in some of the case studies. Another advantage is we can use the automation to use a low amount of material. So hundreds of conditions can be screened in one day as part of your DOE designs, and we can work with microgram to milligram quantities of API depending on the molecule. The data integration into one database is very, very helpful in the sense that we can take information from disparate systems and software and instruments, and all of that data will then go into one database. And I'll show a little bit how that works. But effectively, when you've created this high throughput design, now you have a lot of data coming out, and this shared database allows for the scientists and their managers to very quickly look at the analytical results, including some of the DLS results, which I'll be highlighting today. And we can trace that back to the initial compositions, all the processing, and all the analytical information. And I'll talk a little bit more about how that looks later. And as I mentioned before, this more efficient use of valuable scientist time means that the automation does the tedious tasks, the strenuous tasks, the physically demanding tasks, and the scientists can focus on implementing true quality by design strategies, designing their experiments, analyzing the data, and further iterating through the studies to create very well-designed products and safe products. So how do we do this? For these two case studies I'll be talking about today, we used an automation system that we call the Biologics Formulation system. And what this system has, it has multiple capabilities, and it enables many parts of formulation development to be automated and fully automated. So we start with a HEPA enclosure, which creates a low bioburden workspace and effectively an ISO 100 workspace, so low particulates. And then we have multiple capabilities. On the core module 1, this robot on the right side, we have formulation, analytical, sample prep capabilities, and then we have some of these integrated analytics that I'll talk a little bit about later. In the center, we have a carousel that has capacity for putting pipettes, plates, vials, etc. And this system also allows the different microtiter plates and vials to be passed from the right robot to the left robot, and there you can see the integrated analytical capabilities. And nestled there is the Wyatt DynaPro 2. And as you can see, there's a multitude of different integrated capabilities that the system can implement. And I'll be talking about 2 case studies today that show many of these different capabilities. So when we're implementing these different studies in the laboratory, we have a sort of very high-level strategy. It starts with the design of the experiments, then we go into execution of those designs. We analyze the data and we report the data. So the scientists can, let's go back this way. The scientists then plan their experiments. They can do that at the robot, at their desk, or they can even remotely log in. And they can take designs from DOE software such as JUMP, Design Expert, Spotfire, and whatnot, and quickly implement those into designs that go into the Oracle database, which then speaks to an application server. And I'll talk about how the application server works in a moment. So once those designs are brought in, the scientists can access those designs for a multitude of different workflows. Once those designs are implemented and reviewed, those instructions can be sent to the automation, and formulations can be prepared, they can be stressed, and then they can be sent off into execution mode for third-party systems as well. Then in that initial design, whether it was imported from DOE or it was designed ad hoc, those instructions go to the various analytical instruments. In this case, we can see the DLS is one of these instruments. So the same LIA software can basically send all of these instructions out to these different third-party instruments, whether it's DLS, UPLC, HPLC, etc. The instructions are sent to those devices, and then they collect the data. And the information, let's say, for example, from the DLS, then once the system is done collecting the data, it's sent back to the application server, and all of that data now is in one shared database, linking all of the results. And it makes it very easy for them to analyze and then report the different results. And what's important here is we can actually close the loop. We can take the results from these different experiments, export those to DOE software, and we can take advantage of the very powerful statistical tools that are available in many of those software packages. And so how does this look? So here's an example of when we're querying the database. We can see on the left-hand side here there are unique identifiers for each of the vials or wells in a microtiter plate. And we can see as you go across the row, you can see for that particular unique vial, we have the HPLC chromatogram, we have the gel image from gel electrophoresis, we have the bioanalyzer trace, et cetera, and you can also see the DLS data is here. Now this works somewhat like a web browser, and we can double-click on any one of these pieces of data that are processed, and we can quickly look at the raw data and reprocess that information, and that becomes a new experiment, and it would just become a new column here. So you can see how this is very helpful for scientists and their departments, because they can see very rapidly all of the different data that were ever generated from this original vial, and they can even go back to the very original compositions of those vials. So today I'll be talking about how we can show the value of automation for drug development. And the way we're going to do that is show case studies that demonstrate how when we automate formulation development, how we can see real benefits. So I've split up into two case studies today. One is indicative of an earlier phase formulation study. That's the plate-based. It's a high-throughput formulation screening. And then we have case study 2, which is more indicative of later stage where we're starting to look at devices. In this case, we're going to be looking at serum vials and how we can actually do stability studies in serum vials. So with case study 1, the procedure is very straightforward. We start with using automation to prepare a diverse set of formulations. So in this case, we have air displacement and positive displacement pipetting capabilities. That means we can do low viscosity or high viscosity solutions and liquids and move those into a 96-well plate and create very quickly a diverse set of formulations. We can also make multiple 96-well plates serially. The second part is we can stress those formulations. In this particular case, we're actually going to use the DLS to do stressing, but on these systems we also have vortexing capabilities and we have heated capabilities, so we can have multiple plates at multiple conditions all in parallel. And then we can collect the data, and as I mentioned before, as we collect the data, it's immediately shared it's saved in a shared database, so all that data is linked. And for this particular case, we're going to be relying heavily on turbidity and DLS to not only analyze but also to stress our samples. And then later we can select the robust formulations that will be further analyzed with SE-UPLC. So in this particular case, we start with just making 4 different solutions and putting them onto the robotic deck. So that means the acidic forms of acetate and basic forms of acetate, and same with histidine. And when we mix those at the appropriate ratios, we can create very rapidly a diversity of different buffer pHs at the same concentration. And we can then use the liquid handling device to draw those materials, automatically reformat it to a 96-well plate, and now with dilutions we can create 48 different buffered dilution buffers. And when we add our API plus 2 different excipients, we can very rapidly create 96 different formulations. Then we have the robotic system can pick up this plate and move it to our UV spectrophotometer, excuse me, and then we can collect turbidity data. So this is absorbance at A350 to get an initial time point. Then we can use the robotic system to draw that plate out of the UV vis and move it into the DLS. And the DLS actually does 2 things. It will start by collecting dynamic light scattering information for every well, and then we go through a temperature ramp where we start at room temperature and then we step through until we get to 68 degrees Celsius, and we collect DLS data at each one of those time points. And in the end, we recollect the turbidity data to see if any of the formulations have become turbid as a function of heat stress. And then we can select those, in this case, if the formulations are not robust, then we just remove them from further analysis. If they are robust, then we can move them forward to the more time-consuming SE-UPLC method. And here's an example of what some of that data looks like with the LIA software. We can see on the left-hand side the before and after DLS temperature ramps. The A350 is low initially, so low turbidity or no turbidity in all of the different samples. After the samples have been stressed in the analytical device, we can see that there are multiple formulations now that are highly turbid and then would therefore be removed from, in this particular workflow, removed from further analysis. We also have these abilities to make multivariate plots, so you can see the radius is on the left, and then we have temperature as a function of temperature on the bottom, and we can have a color map here for pH, and we have different ways of looking at the data points to help suss out what are the differences. And this is something that can be configured by the scientists and their teams. And what's important to show here is that we selected only those formulations that are robust, and then we performed then the SE-UPLC to verify that there's no presence of aggregates in those. So this case study is showing the benefits of what we call the BFS and the LIA software. So using automation and using the system to perform otherwise menial manual tasks, we can have much more efficient workflows, and we can reap many of the benefits. So we can use a small amount of API because the system is set up to make multiple 96 well plates at low volumes, and we can use this material, the same material that we use in one plate, we can subject to 3 different analytics. We can create this rapidly. We can create a diverse set of formulations from sterile stocks in a low bioburden environment. And so this is something that would normally take time and is tedious for the scientists to do. This is fully automated in this workflow. As part of that, we can examine the stability of a large set of these formulation variants, which enables better implementation of DOE experimentation and provides the development teams with more information about their product. We can then use this data, this characterization data, to select which of the formulations would justify further testing. In this particular case, we were focused on workflow. In some cases, if you want to employ a true DOE design, you may just want to go through and analyze all of the data, and you can do that as well. And you can even select in real time which samples you would like to run and which you would prefer to leave out. We can aggregate the data and generate customized reports. Once those report definitions are created, they can be locked, and then you can use those over and over again, share them across different development teams, and this enables informed decision-making for the products that you're developing. In case study 2, we were looking at vial-based formulation screening, and this is something interesting because we wanted to look at how vials might be able to be investigated earlier in formulation development programs. This study is very similar to what we did in the previous study, except in this case we're preparing 40 different formulations instead of 96 formulations, and we're preparing them in serum vials, and we're using the robotic system to prepare those. We are going to stress the formulations, but in this case we're actually going to use some of the systems and capabilities of the BFS to stress the formulations, and we're also going to do freeze thaw off the deck. We're going to then collect the analytical data, and we had the reports generated just like we did for the previous study. So in this case, you can see that there's 24 different vials. These pie charts actually show you precisely what was added to each vial to create a unique formulation, and there were 2 of these to make 48 different formulations. These were then capped and sealed. We used the visual inspection station, which automates color, turbidity, and visible particle quantitation. Then those vials were opened, and the liquid handling capabilities were used to draw material out of the vial and then put it into a different format. Now it's a 96 well format. This is a deep well plate. The deep well plate is then subjected to viscometry. In this case, we actually use a microcapillary system, which can be complementary to the DLS viscosity measurements that Dan had just spoken about. And now we can also perform pH measurements in high throughput mode using a 4-tip pH probe. This is a solid-state pH probe, and there's an automated washing and drying protocol for that. So all 3 of these are techniques that are high throughput and unique to the BFS system. And we have now we can take the material and create blanks and samples for the UV vis. We can either make 2 different plates, or we can conserve material and use 1 plate to do both the A280 and if you want, you can do the A350 as well as the DLS. And we can also take material either from one of the original aliquoted plates or from the original vials and start to perform SEC. Again, we can select from the best performers early on to minimize the amount of downstream processing and analysis, or you can process all of them. In this particular case, I wanted to just highlight some of the DLS data. For this particular study, we noted that particle formation was evident in the lower pH formulations. We can see here that we have 4 different points. We have the T0, so the unstressed material in red. In green, we have the freeze thaw, which was done off the deck. But the agitation in purple and the cyan data, which is heat treated, those latter 2 were actually stressed on the system, on the BFS system, in parallel. and then we analyze those materials with the DLS. And we can see here that the pH formulations at pH 4.5 to 5 had higher percent polydispersity values. In some cases, the system was just not able to even calculate the polydispersity. So this was a good indicator of stability for the different formulations. So overall, what we see from this study is that automation is increasing the efficiency of the study. If we look back at trying to perform these manually, we can see something like a 4 to 6 times more efficient program than what we use currently. And what's important to note there is that for this entire study, for all 4 stress points, or for T0 and the 3 stress conditions, it took about 28 hours of attended labor for one scientist to do this entire study. What's also important to note here is when we use the system to make the formulations, we're able to make about 200 vials filled with unique formulations in about 8 hours. And the idea here is that we have only about 2 hours of that is attended labor, and the automation is performing much of this physically strenuous work. So the parts where people would be hunched over in a laminar flow hood pipetting things, the system is performing all of that. And the scientists then can focus on getting their data ready and getting their systems ready, analyzing and reporting. The preparation and analysis time, including all the analytics for any one time point, is about 7 hours for one scientist for 48 formulations. So this is something that's really able to empower the scientists and the teams to do far more work than what would be currently possible. So the advantages, we think, are relatively clear. We can see that with the ability to test the systems and calibrate the systems, there are opportunities for improved results for each study. We do see a significant productivity improvement. We can lower the amount of material that can be consumed by using multiple analytics for a given amount of material. The data integration into one shared database is very powerful. It makes it very easy for scientists and their teammates and even across departments to look at what they've done and very quickly see how the different data relate to each other. And this can enable decision making. It can really provide good understanding of what was done and how the products are reacting to the different conditions. And what's really important here is now the scientists can take their time that was normally previously used for doing manual, tedious methods, and they can take that time and use it to refocus their efforts on implementing quality by design strategies, meaning making very well thought out DOE experiments that can be iterative throughout the process and with the intention of shortening product development times. So I'm just going to show here very quickly as I wrap up. These are the different formulations that we looked at here so that you can see this. This is available on our website as an app note if you'd like to see more. And there is additional information available on our website in our Learning Center. We have multiple webinars and other information to talk about how these systems have been used in the formulation development, not just in biologics, but also small molecules. So I want to thank you for your attention, and thanks to the teammates that helped me with this work and the presentation.
Thank you very much, Russell. And to conclude the webinar today, we will now have a brief question and answer session. So the first question is for Dan. Is it possible to determine multiple conformation or unfolding changes in a single solution? Is this orthogonal to data obtained via differential scanning calorimetry?
Regarding the first question, different conformations will not be vastly different in size, and the resolution of dynamic light scattering, in order to actually get different peaks in the multimodal example that I showed, is something like a factor of 2 or 3 in radius. typical unfolding of a protein is not going to lead to that large of a size. And what will lead to is increase in polydispersity, so that the width of that peak will increase so much. But if you have, say a folded conformation and unfolded conformation, you'll see that increase of width. Different folded conformations wouldn't be too different, and so you wouldn't get a lot of discrimination. It is definitely orthogonal and nicely complementary to differential scanning calorimetry, because DSC tells you that something is happening. You see evidence in the calorimetry that some transition is occurring, but you don't really know what it is. The dynamic light scattering, because it's giving the measurement of size, will tell you if it's unfolding and aggregating more directly. So they're nicely complementary.
Okay, and the next question is for Russell. How does one make sure that mixing of viscous solutions in the small volumes is complete when using a robotic system?
So the way we do it is we'll add the solutions together, and we have the choice, as I showed previously. I think I'm still sharing my screen, so maybe I can even just go back to the deck element here. We can actually mix those solutions as they're being added, and here's the vortexing orbital shaking. You can actually put your vials in these positions with the vortexing and orbital shaking, and you can go from 0 to 1,000 RPMs, and you can either add all of your materials and then mix, or you can, as you're adding the materials, mix them to ensure that the solutions are properly mixed. You can also do tip mixing. So if you're using the Eppendorf tool, which we have, you can actually use the Eppendorf. You can see the Eppendorf. In the end, you can grab a clean Eppendorf tip if you'd like, and you can draw all of that material into the Eppendorf and then expel it, and possibly do this a few times to also mix. So there's various strategies for ensuring proper mixing.
Okay, and the next question is actually directed to both of you. Have you compared any of the results gotten from the automated system to a non-automated system?
I can take this, Dan.
Okay.
So, and let's. Well, I guess I can answer first, and then Dan can answer as well. So because it sounds to me like that's geared a little bit more towards automation generally. So we do have some information on our website. I showed the link earlier. We are working on a paper right now with a biotechnology company where we are showing we are showing just that. We've conducted the same studies that they conducted. The only difference was we did everything in automatic fashion or largely automated fashion, and they performed everything in a largely manual fashion. And we actually found that we had the same results. And so we're working on that publication right now. And as I mentioned, we do have other information on our website which does support some of these additional claims.
Just in case the question is asking about dynamic light scattering in the plates versus in the standard cuvette format, of course, Wyatt offers both. And in the quartz cuvettes, you get slightly better sensitivity, not vastly different. You can run over a much larger temperature range. So we can run quartz cuvettes up to 150 degrees if you'd like. The temperature range is not as much as in the plate grade. It's only up to 85. But otherwise, the results are very similar.
Okay. And this question is for Dan. Can you discuss the concentration sensitivity when doing KD measurements? And do we need to correct for viscosities?
Oh, that's a question which is probably on the cutting edge and still under discussion. If the viscosity is due to the solvent, so for instance excipients that you have in a solvent, then that's something that you definitely want to put into the analysis. Otherwise, the typical KD measurements are made at relatively low concentrations, say up to say 10 or 15 grams per liter, and that's in order to remain in the linear regime. If you remember the equation I threw up there, the first, it's a series expansion concentration, so the first order term is the KD. As you go to higher concentrations, you'll have C squared terms and CQ terms, and so, if you're going 40, 50, 100 mg per mil, you can't just use the first order expansion. There have been publications, and there's actually one referenced on our website where people have looked at high concentrations of proteins with dynamic light scattering in order to get a rough correlation, but it would be very interesting to have some more extensive studies to understand how the diffusion coefficient changes with concentration at these very high concentrations correlated with static light scattering studies, which are theoretically better understood. That is still lacking in the literature.
Okay, and this question is for Russell. What's the sensitivity of using UV-Vis plate reader to determine turbidity in comparison to a nephelometer?
That's a good question. I actually don't know the answer to that. I do know that we are using the SpectraMax and the M2, and so that would be part of the specifications for that particular system. So I know we can see values down to a tenth of an absorbance unit at A350. I think that still would be meaningful. So, how that compares to a nephelometer, I'm not exactly sure. We also have, as I mentioned before, the automated visual inspection system that also can look at turbidity, and we can compare turbidity standards that are, we have accurate quantitation even down to about 2 to 5 NTU with that system. I would suspect that the SpectraMax is even far more sensitive than that. So if you're looking in the even 0 to 2 NTU range, the SpectraMax is going to be very helpful for that. And above that, the visual inspection station can go all the way to 800 NTU and can very clearly see the differences. So there are, in this particular automated system, there are ways to look at both of those regimes, the very low turbidity versus the very high. But how that reflects and is comparing to your nephelometer, I'm not sure. But maybe with that information, you could make some comparisons.
Yeah, just briefly, turbidity is measured in a few different ways. So it can be measured by transmission, it can be measured by scattering. The scattering will depend on the wavelength, also depend on the distribution of particle sizes. So each of them give you slightly different representations of turbidity, and there's no hard correspondence between any two systems, I believe.
Mm-hmm. Thanks, Dan.
Okay, well, thank you both. We are about out of time and must conclude our webinar today. Many great questions have been submitted, and though we haven't gotten to all of them, we will compile a written response with all the questions and answers, regardless of if they were read aloud. We'll send you a link for the archived version of this webinar and Q&A document to view on either the Freeslate or Wyatt websites. Wyatt's on-demand webinar library is also a great resource where you can find recordings covering many additional aspects of light scattering technology and applications. If you have further questions, please get in touch with me at the contact information in your webinar confirmation email. Thanks again, everyone, for joining us, and a big thank you to Dan and Russell for your presentations. And this now concludes our webinar for today.